In this video, we are going to talk about the differentiation of bacteria based on the staining of the cell wall. The cell wall is a protective layer located outside the cell membrane of the bacteria, and the cell wall is composed of peptidoglycan layers. The peptidoglycan is the combination of glycans and peptides. In another video, I have already explained the structure and synthesis of cell wall. You will find it on my channel. Based on the staining of the cell wall, the bacteria are classified broadly into two types. In 1884, a Danish scientist named Christian Gram developed a fundamental technique that classified bacteria, and that is called Gram staining or Gram's method. By the way, his technique was not intended to do the classification, rather he was trying to make the bacteria look more visible by staining method. It is actually some coloring methods. And as a bonus result, all the bacterial species were classified into two groups: the gram-positive bacteria and the gram-negative bacteria. Okay, so let us see the differences. So here we draw the gram-positive and gram-negative bacteria. That is the general structure with the plasma membrane, genetic material, and ribosomes. The fundamental difference between them is in the layer of the cell wall of bacteria. Gram-positive bacteria is composed of multiple peptidoglycan layers, which forms a thick cell wall. The thickness of the cell wall is around 20 to 80 nanometers. But gram-negative bacteria is composed of a single peptidoglycan layer which forms a thin cell wall, and that is around only 10 nanometers. But the cell wall of gram-negative bacteria is actually more complex than the gram-positive bacteria. In cell wall, there are peptide linkages. As the layer of gram-positive bacteria is thick, it makes some network rich in peptide linkages. And those linkages make the cell wall of bacteria kind of porous. It means some pores may be produced. But as the cell wall is a single thin layer in gram-negative bacteria, it does not make any pores like gram-positive bacteria. These pores actually help the bacteria in transporting necessary proteins and nutrients for the survival of bacteria. Now, there is a question. The negative bacteria do not have these pores, so don't they need to transport anything? Alright, just hold on a moment for getting this answer. Now, a gram-negative bacteria consists of an additional outer membrane, and this outer membrane is composed of an extra phospholipid bilayers, extra proteins, and a special structure that is lipopolysaccharides. But gram-positive bacteria do not have any extra layer. You see, the positive bacteria has thick peptidoglycan layers, and there is not really too much free space. But due to the thin layer in gram-negative bacteria, it has too much free space to hold an extra layer. And this extra layer provides highly protective barrier for gram-negative bacteria. Now, let's draw a part of this bacteria in more detailed view. The phospholipid bilayer, the cell wall, the peptide linkages, and the proteins on the lipid bilayers are common for both the bacteria, and this is the outer membrane of gram-negative bacteria with the extra phospholipid bilayers, extra proteins, and the lipopolysaccharides. So you see, due to the extra phospholipid layers on the outer membrane, the gram-negative bacteria becomes more concentrated with lipid compared to gram-positive. So lipid concentration is less in gram-positive bacteria. Alright, now there are some protein channels on the outer layer of gram-negative bacteria. Through this channel, necessary ions and molecules can be transported. These are called porins. Since gram-positive bacteria do not have extra layer, they do not have porins. However, they do not need it because for transporting molecules, they already have some pores created by the peptide linkages. So we got the answer, right? How the gram-negative bacteria is able to do necessary transportation, that is through the porins. Because of the differences in the thickness of the cell wall, the periplasmic space between the plasma membrane and the peptidoglycan layer is really thin for gram-positive bacteria, and it is thick for gram-negative bacteria. The periplasmic space is made up of a gel-like matrix that basically consists of various types of proteins concerned with cell's metabolism. Gram-positive bacteria contains a teichoic acid and lipoteichoic acid molecule. These are basically copolymers of glycerol phosphate or ribitol phosphate. The teichoic acid is directed from the peptidoglycan layer, and the lipoteichoic acid is directed from the lipid bilayer, and these carry the antigenic property of gram-positive bacteria. Gram-negative bacteria does not contain this teichoic or lipoteichoic acid, but the antigenic feature of gram-negative is carried by a part of outer membrane. Both the bacteria can produce toxins. The toxins produced by gram-positive bacteria is exotoxins, that is released by the bacteria into its surroundings. And gram-negative bacteria also release exotoxins as well as endotoxins. The endotoxin is a toxin that is present inside the bacterial cell wall and released when the cell wall disintegrates. You will get a video on exotoxins and endotoxins of bacteria on my channel. So exotoxins are secreted and released by the bacteria, but endotoxins are integral part of gram-negative bacteria. That is actually the lipopolysaccharides in the outer membrane, and it becomes the endotoxin after the cell is destroyed. Now the Gram staining method. It is a stepwise technique that differentiates the bacteria into gram-positive and gram-negative groups. The gram-positive bacteria in Gram staining method appears as purple color under the microscope, but gram-negative bacteria appear as a pink color under the microscope. There is another video on how the Gram staining technique is done and why do they differ in their appearing colors. Now, one more thing, the gram-negative bacteria are more pathogenic than gram-positive bacteria. Gram-negative bacteria have been developing dangerous resistance compared to the gram-positive. We all know what are superbugs. They are powerful bacteria that are resistant to the antibiotics available for treatment. And most of the superbugs are gram-negative bacteria. And that's why the gram-negative bacteria are classified by the Center for Disease Control as a more serious threat for us. The examples of some gram-negative bacteria are Escherichia coli, Pseudomonas aeruginosa, Neisseria gonorrhea, Helicobacter pylori, Klebsiella pneumonia, Haemophilus influenza, Meningococcus rickettsia, and so on. And some of the examples of gram-positive bacteria are Staphylococcus aureus, Streptococcus pneumoniae, Mycobacterium tuberculosis, Bacillus cereus, Clostridium botulinum, Clostridium tetani, and so on. Alright, so these are the major differences of gram-positive and gram-negative bacteria. I hope you understand all the differences, so like this video and put a comment if you have any question or anything to say. And obviously, don't forget to subscribe. Thank you.